Hello, I am Pierre Bellec, and today I'm here to tell you a little bit about brain parcellation in the courtois.neuromod 2020 release. So, the main objective of this work is as follows. We want to generate brain parcellations. Brain parcellations are like a map of the brain into little regions, and those regions are defined so that they are composed of brain parts that have similar function. And in the literature so far, that's been looked into a very limited number of cognitive domains. In the courtois.neuromod project, we have a lot of individual data covering many different cognitive domains, whether those are movies, TV shows, or different types of tasks. So our objective today is to build high-quality individual brain parcellations, which are going to generalize to a variety of videos and tasks in courtois.neuromod 2020. Or in other words, one parcellation to rule them all, and in the embedding space, bind them. Before we start, I would like to acknowledge the people who contributed to this work. I did a bunch of the coding, but this is a continuation of an algorithm that was developed by Amal Boukdir during her PhD thesis under my supervision, the supervision of Max Mignot. The bulk of the heavy data processing was implemented by François Pogam and Anat Chamarquet. Valentina Borghesani is also in the team looking at annotating the parcels. And finally, Dr. Yu Zhang also helped direct choices we made in the design of the algorithm. I would also like to acknowledge the entire CNeuroMod team and all the people who get scanned and have scanned them, which was a tremendous amount of work, and also the sources of our institutional and funding support, and in particular la Fondation Courtois. So now let's get started with our background and objectives. So why use brain parcellation? The Courtois project is really about training artificial neural network to imitate brain activity. And so far in the literature, when people have really tried to discuss whether artificial neural networks do the same thing as a biological neural network, a brain, they've done so by putting in correspondence some layers of the artificial neural nets, which we see here at the top. Gosh, my hand is getting cut. This is terrible. Not a problem. Now I'm big. Here, those layers of the artificial neural net, and try to relate the activity of these artificial layers to parts of the brain which should correspond to the same degree, let's say, of abstraction of the features extracted here from an image. And to determine which part of the brain should support this type of abstraction representation, people have this type of brain parcellation. Here the visual areas 1 to 4 and the inferior temporal cortex, which are known to create a cascade of representation of visual information. So, how do we get those parcellations so we can extend this type of approach to the full brain? That's the big question we have. So let's talk a little bit about how parcellations are typically generated in fMRI. Well, the basis for those parcellations are what's called functional connectivity. So it's a phenomenon known since 1995 and the early work of Bharat Biswal. If you take a particular point in the brain, so here we've got the posterior cingulate cortex. Sorry, I'm clicking away too much. The posterior cingulate cortex, here shown in a rostral view. You have the other views here, sagittal and axial. Well, you have a spontaneous fluctuation in that brain region, even if the subjects don't do anything in particular. That's shown here. And then we can correlate this spontaneous activity, or this activity even during the task, with the activity seen in the rest of the brain. And what we're going to see is that there are parts of the brain which have highly correlated activity. Typically for individual subjects, we are looking at correlation above 0.5, but that can reach very high level, 0.8 to 0.9. And those correlations are distributed along what's known to be regions connected through neuronal pathways. Here you have a famous network, the default mode network, and those regions are all connected to the posterior cingulate cortex and are involved in specific types of information processing in the brain. So we're able to create a map of that network just by looking at the coherence, the temporal synchrony of activity between regions in really any kind of cognitive context. So that particular method is pretty crude. You need to select a seed and you get a map. You don't really get a parcellation per se, but, and let me get rid of myself. So you can use data-driven cluster analysis to automatically detect those type of large-scale network I showed on the previous slide and create a real map of the brain. Those maps are just like integer values. You're going to be in parcel number 1, parcel number 2, parcel number 3. The number of parcels is more or less arbitrary. The truth is that the brain has a multi-resolution structure, so you can make maps that are at a very low level of detail. Think about maps of the continents of the Earth, or maps with a higher level, let's say maps of countries, but then you can go to provinces and towns and whatever. It's the same for the brain. Another little important point I'd like to stress is that in the literature, there's two big kinds of parcellations that have been considered. At the top, you have the Yeo-Creenan parcellation with two different numbers of parcels, 7 and 17. Those are really exactly like countries, right? Like you are in the country or you're not at the country. There's a frontier, and every place on Earth is in one country and one only. Those parcellation work that way. At the bottom, we've got what's called soft parcellation. And those parcellation have kind of a weight. So you are more or less in a country, if you wish. And also there can be some overlap. So some parts of the brain may belong to one parcel with a certain weight and another parcel in another weight. Sometimes I refer to this type of parcellation to dynamic parcellation, because the truth is that for a given voxel in the brain, over time, its strength of belonging to one parcel or another can change. And so you can really capture with this type of parcellation dynamic reconfiguration of brain parcel, which is not possible with the top type of parcellation. So hard parcels, soft parcels. The bottom ones are the Diffumo parcel created by a team in INRIA in Paris, the parietal team, which are currently some of the highest quality parcellations that have been generated. And I'll discuss a little bit more about this in a minute. So how do we decide that the parcellation is a good parcellation? Well, there's a couple of criteria that people have used. And the first one really is reproducibility. So here what you have is two subjects. And for each subject, we have two data sets that have been collected independently, roughly 3 hours each, a discovery data set, a replication data set. And so what you have is, both discovery and replication parcellation for both subjects. And as you can see, this is very, very, very reproducible, right? Like if you look at a lot of those details, like the visual network here and there are almost exactly the same. They're slightly different from the other subject, but for the observed subject, it's also perfectly reproducible. That's great. Now, if you zoom in, there are still some little variation, even though we're using 3 hours of data to generate those parcellation. Xu and colleagues from Thomas Yeo's lab use 3 hours. Here and there in the posterior cingulate cortex, that region I was talking about for the default mode network, there are some slight differences in shape, even though the solution have been generated from a large amount of data. So how much data do we need to really get reproducible individual parcels? Well, here are some graphs from the laboratory of Katrina Gratton. So on the left, you have the reproducibility of functional connectivity map as a function of the amount of time used to generate those maps. So those are like the maps of the DMN I was showing you at the beginning. And as you can see, even with 10 minutes of data, you have fairly high reproducibility, 0.7 on a scale that goes up to 1. And if you continue extending after an hour, you reach an asymptote above 0.9, which is considered excellent reproducibility. Now, if you're talking about the parcels, which are really more like binary version of those maps, then things are not looking that great. With 10 minutes, you're at only the dice, which measure kind of an overlap between test and retest, you're at only 0.5. And even if you pour 2 hours of data, you're reaching this like very slowly an asymptote that seemed to be actually 0.7 or 0.8. So with Amal booked here during our PhD, we're trying to get that to become better. And that was one of our core objectives. And we really struggled getting there. She developed a new approach for dynamic parcellation, which we believe can help with that. And that's one of the things we're going to explore in this project. Now, I just told you about reproducibility. Let's move on to our other main metric for quality, which is homogeneity. In the simplest case, homogeneity is simply kind of like the correlation of the activity of each little unit in a parcel with the average activity of that parcel. So whether the average activity of a parcel is a good proxy for the activity of everyone inside. So here's a little graph representing many different parcels that have been proposed in the literature, including random ones. Those are the red dots. And we have our Yeo-Creenan parcels. They are at the bottom here with 7 clusters and 17 clusters just above. And on the x-axis, what you have is the average size of a parcel. On the y-axis, what you have is the homogeneity. And as you can see, there is a very tight relationship between the size of the parcels in the parcellation and how homogeneous they are. In other words, if you look at a map of towns, each little unit in your map is more homogeneous than if you look at maps of continent. Surprising? Not exactly. But here are the numbers. If you use a very low number of parcels, very big parcels, you at only 0.2 correlation with your average, which is really low. And even if you look at hundreds of parcels, like the Gordon parcellation that has small parcels, you still cap at around 0.6, which is still far from perfect. Now, one of the limitations of those parcels is they're all hard parcellations, right? Like the country. So that's kind of a limitation to really approximate the activity of the brain, provided if there are some dynamic reconfiguration, right? So let's see how it works with the soft parcellation. And here we go. We've got the diffumo parcellation I told you about a little bit before from Daddy and colleagues. And what you can see here on the left is a measure of homogeneity for soft parcellation. And here what's being done is that you take a map of brain activity and you compress it, you embed it in the space of this parcellation, and then you rebuild an approximation of that map. And you measure the quality of the embedded map compared to the original map. That gives you what's called an R-square score, which is on the y-axis of the graph. Let me get big. On the y-axis of that graph. I love being able to change size like that. Sorry, I'm having way too much fun. So on the x-axis here, we've got dimensionality, the number of parcels. So, the higher the number, the smaller they become. We again have a bunch of different parcels, and you can see some are hard and some are soft. And you can see that, again, like the more parcels you have, the more homogeneous they become. But now you can see that diffumo, which is in purple, really outperforms even other kind of popular parcellations, such as the Schaffer parcel that are here, which are very widely used. So that means that soft parcellation are better able to capture fMRI dynamics. And one way of visualizing that is to look at activation map, a little bit like what Valentina presented you for the HCP-TRT task. At the top, you have an activation map computed directly from voxel, so with all the measurements units we have. At the bottom, you have a map that's been embedded with hard parcels that were generated in the lab. They're called the Basque Atlas, and they suck. I can say that because those are like that. I developed this algorithm. But in general, hard parcels, they don't do a really good job. And in the middle, you've got the diffumo parcel, and you can see that the approximation is beautiful. It's almost look like the real thing, even though here we only have 1,000 parcels, while at the top, you have tens of thousands of voxels. So great reduction in dimensionality, not a great loss in terms of precision. On the y-axis here, we see we cap at around 0.6, but now we're in R square. So 0.6 is, R square is much higher than 0.6 correlation, if that makes sense. Moving on. The last thing I wanted to tell you to introduce you the context of the project is generalization. So another kind of key point in the literature is that those parcellations are often generated while people don't do anything during what's called the resting state, or spontaneous brain activity. But do the parcels really depend on what people do? Some people don't think they should, because they think those functional parcels, they mainly reflect how the brain is wired, which doesn't change that much from what you do from one task to another. But Saleh and colleagues from Todd Constable's group in Yale, they showed that actually if you compare parcels that have been generated from the same tasks, that's in orange, for single and individual. In orange, you compare them from the same task, you have very high overlap in the 0.8, which is actually higher than what I was showing you from the Graton's lab. But if you compare them across tasks, then you go down to 0.7, 0.75, which was where in the work of Katerina Graton we were. And that's a replication in a different data set, actually, that the Midnight Brain Scan data set, which was used in the work from the lab of Katerina Graton. So it seems that there's this robust findings that hard parcels really depend on the tasks that people do. But maybe it's just because they fail to capture those dynamic reconfiguration, which are different in one task compared to another. So let's give now all the objective for today's talk, the detailed menu, if you wish. Which brings us to our specific objectives and hypotheses for this study. So our first one was to extend the algorithm for soft parcellation that was developed by Amal Boukdir during her PhD to the full brain. Our original work was based on SEED, Foxel. We hypothesized that this method could scale to a very large fMRI dataset, and also, just like DFUMO, could improve on the quality of the embedding of fMRI data. Our second objective was to assess the reproducibility of those parcels across different cognitive contexts. So extend the work of Saleh and colleagues, but instead of just looking at control task, also looking at many different types of videos, including different kinds of movies, as well as some TV shows. And finally, we wanted to assess the homogeneity of those individual parcels across different cognitive contexts. And as I hinted at a little bit earlier, we think that because the dynamic parcel capture different states, even within a given context, hopefully most of those states will be reproducible across context. And so we're hoping that the homogeneity of our parcel is going to be comparable across different cognitive contexts. Now let's go to the method and see how we tested those objectives. Methods. So we are using the CNeuroMod 2020 release. We have the movie 10 data, which is about 12 hours of movies. And those movies are Born Supremacy, Wolf of Wall Street, Life and Hidden Figures. Life and Hidden Figures have been repeated twice. We also have the Friends TV show that those guys, we watched, the participants watched 2 seasons, which total about 20 hours of data, 18 exactly. And finally, we also replicated the task battery from the Human Connectome project. And those tasks involve 8 domains. So you've got them listed here. You've got the gambling, the motor, working memory, social language, relation, emotions. Those are the 7 main tasks. But we also have some resting state, which was used a lot prior to generate parcellation. Now, the difference with HCP is that in HCP, each participant repeated those tasks twice, and in CNeuroMod 2020, they repeated 15 times. So for each subject, we have almost 10 hours of data. In total, we're looking at roughly 40 hours of data in different cognitive contexts per subject. And we have 6 subjects, 3 men, 3 women. So that's something like 240 hours of fMRI data we're going to be playing with, high temporal resolution. So in terms of the pre-processing, fMRI data was pre-processed with fMRI prepped. fMRI prep, the 2020 long-term service release. All the details of the pre-processing steps are in our technical documentation, which I've linked on the slides and will share the slides. fMRI data was resampled in the MNI 2009 template developed by Fonoff and colleagues, team of Louis Collins. 2 mm isotropic spatial resolution, which was our acquisition resolution. We found that the parcels work better when generated with quite an aggressive smoothing at 8 mm. But in terms of measuring homogeneity, we wanted to have a regime that's closer to the real resolution of the data. So we assess homogeneity at 5 mm isotropic. Finally, in terms of denoising, we've used the PAM36 strategy, which was proposed in Cyric and colleague in 2017, using the library developed by the lab that's called load confound. Now, in terms of the DIPAC algorithm, how does it work? Well, there's like 2 steps. First step, you take your fMRI time series and you cut it into small windows, small chunks. There they are. Let's say typically for one run of fMRI, we have about 400 time points. So we're going to use 100 different windows equally spaced in our time series. For each of those windows, we're going to generate a parcellation, hard parcellation, classical, boring. Actually, we use the k-means algorithm, which is one of the oldest algorithms to do that. Now, instead of representing our parcels with little colors in the brain, like I've shown you so far, what we're going to do is that we're going to create what's called one-hot encoders. So we're going to create separate map for each one of our parcels, and those maps are going to be binary. 0 when you're not in the parcel, 1 where you're in the parcel. And now what we're going to do is that we're going to concatenate, put together all those maps, and then we're going to have this gigantic array of maps, millions of maps, actually. I'll get to that in a minute. And we're going to be running a new k-means algorithm, which is going to try to detect reproducible maps, so states of parcellation. They may be in different states because they're just like completely different networks. Like imagine if we were to use different seeds in the brain, or they may be different states for the same kind of network, but a slightly different configuration. And now that we've grouped all our one-hot encoder vector together, we can average them and get a stability map that's presented on the right. And that map goes from 0 to 1. When it's 0, that means that that voxel was never in a one-hot encoder for that state. When it's 1, it was in all the one-hot encoder for that state, and if it was like 0.5, it was in half of them. Well, you get the idea, right? So when we measure reproducibility, what we do is that we actually correlate spatially those parcels, those stability maps between test and retest. All right, so you get the gist of the algorithm. It's a very simple algorithm. It's what's called ensemble clustering because we're aggregating many different solutions to get stable ones. But unlike traditional ensemble clustering for fMRI, here we're allowing to have different states. And so our stability maps may overlap. And they're also probabilistic, so they're soft. So really, this algorithm, which we call DIPAC, is a soft parcellation algorithm, just like if you will. Now, let's talk a little bit dimensionality. Here are our time series. In the gray matter, you have about 150,000 voxel, but let's round it up, 100K, here, on the left. My hand disappears, that's awful. Oh, here it comes back. Good, so we have about 400 time points. We're going to extract 100 time windows, right? So we have 100 of those cluster maps. We are going to just like put them into a column of one-hot encoder. So we are going to get one different column for each cluster. Let's say here we do 1,000 different clusters, 1,000 different parcels. We're going to get a matrix, which is 100,000 voxel times 1,000 parcels. That's for one window. But this matrix is sparse. Most of its coefficients are 0. Why? Because, you know, the parcels are non-overlapping. So if you have a 1 in a column, all the other columns will be at 0. So clearly that matrix is filled with 0, and we encode it with a sparse representation, which is very efficient when there's a lot of 0. So really one of those matrices is only roughly 100 kilobytes, which is tiny. Moving on. Now, we're going to repeat that. Here's time on my head. Here are many sliding window, 100 of them. For each one, we create one of these sparse matrix, and we concatenate them across all replication. So we still have 100,000 voxels, but now we have about 100,000 one-hot encoders, right? Because we had 1,000 per window, and we have 100 window. And that's still tiny, 10 megabytes. Ridiculously small. But we're going to apply this to an entire cognitive context. So let's say for movie 10, we have roughly 10 hours of data. So instead of having one run, we have 50 of them, roughly. So now we have all our little windows for each run, and we have all our little sparse arrays for each window, and we concatenate all of that, and now we have 5 million of them. And so our one-hot encoding matrix is 5 million times 100,000. That's a lot of megapixels. Like, I'm not even actually risk to say how many megapixels that would be if that were a picture. It's way too much. But it's still not that big in memory because it's a sparse array. So the size is roughly 500 megs, half a gigabyte. So definitely, most modern laptop of like 8 to 16 gigs. So it's still not really a problem to scale to that. So in terms of scalability, let me disappear. The memory footprint is very reasonable and actually doesn't depend on the number of clusters because sparse Boolean arrays works like that. Now, running k-mean at the second level on such a large matrix, which is 100K times 5 million, is actually feasible and only takes a few hours with a couple dozen cores. And the reason is that we use a very high quality implementation by the scikit-learn library, which does support sparse array and multi-core processing. So that implementation is able to leverage the fact that most of the elements in our matrix are zeros, and is also able to take advantage of our high performance computing infrastructure. So in brief, we're able to process the CNeuroMod 2020 data at the individual level without trouble. The implementation of this library was made public already. It is on GitHub. I've put the link here. It's under the courtois.neuromod organization under the name repo DiePack. So now it's time for the results. Let's see what those parcellations look like. Well, first, let's see how reproducible they are. What you have on this side of the screen is a histogram of the reproducibility of the parcels when you're comparing parcels generated on the first season of Friends and the second season of Friends. So for each parcel in the first season, we're going to find the parcel from the second season that has the maximal spatial correlation. And we're going to do a histogram of all those spatial correlation values, which we obtain by matching parcel for the same subject across 2 seasons. And as you can see, most of the values are above 0.9. Actually, the median is typically above 0.95. There's lots of parcels that correlate in the 0.98, 0.99 territory, which is near perfect. The threshold for excellent reproducibility is indicated here. And what the tail is in the range of 0.8, which is generally considered good reproducibility. Here you have our 6 subjects. So you can see that this is that works every time. Parcels with 10 hours of fMRI data are just ridiculously reproducible using DiePack, which is great. Now, on the other side, what you have is essentially the same thing. But now, instead of matching the parcels across season, but for the same subject, we cross parcel across subjects for the same season. And as you can see, for different subjects in general, the parcels only have a reproducibility index around 0.5, 0.6, which is only moderate reproducibility. And the max you can get is around 0.9, right? So that means that all those parcels that are above 0.95 are only seen when the parcels are generated from the same subjects. So we can almost tell who's who just by looking at certain parcels which are extremely reproducible. So we can fingerprint our subject based on the reproducibility of the parcels. Just by looking at those histograms, we can see that it's going to work extremely well. So here what they look like. On that side, you've got parcels that reproduce well, very well. So on this side, you've got season 1. On this side, you have season 2. The first row is actually our top reproducible parcel, which is identical in both cases. That's the only one that does that, but it's at 100% spatial correlation. A very nice little cerebellar region. Here we've got this sweet little frontal area that matches at 0.99, and here another singular area that matches at 0.99. Also notice how those high reproducibility parcels, although they're probabilistic with a score from 0 to 1, they're actually almost binary, which means that those patterns were detected again and again and again on small windows almost perfectly. On the other side, though, here are worse parcels. Here we've got 0.68 roughly spatial reproducibility. So the worst are not that bad. They're almost good actually in terms of reproducibility. And what you can see is that sometimes there's going to be maybe an additional region like here, that thing. Here you have 2 blobs, and here you have only 1 blob. But the blob that disappears had a slightly lower stability. So it was less reproducible in the first place when we aggregated our states. Other maps is just like a slight change in the stability values, like this little blob here has high stability, kind of, while at the other retest, it's very low for season 2. So as you can see, even the low reproducibility parcels are actually quite reproducible when we compare exactly the same kind of stimuli, season 1 and season 2 of Friends. They are not overlapping stimuli, but they are the same actors telling the same kind of story. Now, let's compare that to group atlases. On the x-axis, you have the different atlas, the DIPAC atlas with different number of parcels. So we've got DIPAC with 256 here, 512 here, and 1024 at the very end. And we matched actually the numbers that were used in the diffumo group parcellation. So you've got the diffumo is right there. And as you can notice, DIPAC is better than diffumo. Green versus red. Here at 512, it's pink versus gray. And once again, DIPAC really works much better. And here at 124, it's like yellow versus blue. And again, DIPAC works much better. Now, I believe the main difference, the reason why DIPAC works better is because we're comparing individual parcels with group parcels. I'm pretty sure that diffumo, if it scales to the size of data, which it should also give us fantastic parcels. But really, if we take the best group parcels on the market right now, and we compare it to individual parcels, we see that the individual parcels win. Our homogeneity scores are indicated on the y-axis, and we use the R-square, just like the Daddy and colleague paper. And you can see that we're reaching roughly 0.5, 50% of the variance explained with 1,000 parcel, knowing that our base resolution is 150,000 voxel, smooth with 5 millimeter kernel. Now, there's a few other parcellations on this graph, and the point I'm trying to make with them is that our parcellation just don't embed data really well. So if we look at the MIST atlas, which was developed by my lab with about 200 parcels or 400 parcels here, they don't explain as much variance, nearly as much variance as diffumo with 256 parcels for DIPAC. Or diffumo with 500 really beats all those things. So in a way, we just reproduce the result of the parietal group that soft parcellation embed data better than hard parcellation, but we're adding that individual parcellation embed data much better than group parcellation, provided we're in the same kind of cognitive context as the one that was used to generate the embedding in the first place. One last point is that we can also try to compare the R-square when we use the parcels from the subject in question or a parcellation coming from another subject. So in blue, you have parcellation coming from another subject. In orange, you have from the same subject, and here you have the 6th subject of the study. And you can see that systematically, using the parcel from the right subject gives you a big boost in R-square. The most impressive being subject 3 here that gain almost 15% of variance explained by using an individual specific parcellation. So that's another result that comforts us in this idea that parcels are really subject specific. We saw it spatially, but here we see that this spatial specificity leads us to better compression of individual fMRI data. All right, so now what happens when we try to compare parcellation from different cognitive contexts? So here, that graph I already showed you. We're comparing parcel from the first and the second season of Friends. Here, we are comparing parcel from the first season of Friends and parcel generated on all the movie 10 movies together. And what you can see is that the tail of the distributions for all 6 subjects become much heavier and now goes all the way down to about 0.6, which is only considered moderate reproducibility. That was completely absent here. The tail really almost stopped at 0.8. Here there's a heavy tail for 0.8 and there's a chunk of the distribution that now lives between 0.6 and 0.8. Actually, the lowest value can be as low as like 0.3. So let's have a look at what those type of parcels look like. So we do have some parcels that match really highly between Friends and movie 10. Those are represented on this side of the screen, and we can see really pretty parcels. Another cerebellar region, sensory cortex of bilateral frontal cortices, virtually identical between Friends and movie 10. But if we look at the lowest reproducibility parcels that are presented on that side of the screen, we now see values of matching in below 0.4. And what we can see are things like this cerebellar region, which is very strong, stable on Friends, and which is absent on movie 10. Same here, we have this singular region here on Friends. Again, very similar parcel, but it's missing the singular cortex. So we start to see qualitatively different parcels. And this is not just distributed differences. Here we have an example of a region in the frontal cortex which shifts from one hemisphere to the other between Friends and movie 10. So it's roughly the same location, but the exact border of the parcel will change. So now let's see what's going on where we compare Friends with HCP-TRT, so control task from various domains. And actually, a little bit surprisingly, so here you still have for reference the distribution, Friends season 1 compared to season 2, excellent reproducibility. And on my other side, it's also pretty excellent. There's a more, the tail now extend in the 0.8 range, 0.7 to 0.8 range. So reproducibility that is good without being excellent, but it's actually the tail of the distribution is higher than what we observed with movie 10. So if we look at the kind of reproducibility we can have, we again have those parcels that reproduce really highly. Actually, that frontal one at the very top was also one of our top parcels when Friends season 1 against Friends season 2. It's also identified almost identically in HCP-TRT. And if we go look at low reproducibility parcel now, it's very much the same kind of configuration we've had when we were comparing season 1, season 2 low reproducibility parcel. The quantitative value are a bit lower, 0.55 instead of 0.6 and something. But it's mostly just a slight change in the intensity of the stability of the parcels, more or less. So, spatially, Friends, movie 10, and HCP seem to have very reliable patterns, and sometimes they are those kind of like more outliers that are less reproducible and that are mostly present in movie 10, and a little bit with HCP-TRT. Now let's look at what's happening in terms of R-square, the homogeneity of the parcel. Well, here you've got the distribution of the R-square here on the very left, both for Friends, season 1 and 2. And as you can see, the reproducibility of the R-square is very high. The blue and orange distribution are almost identical. But then when you look at the different movies of movie 10, like Born Supremacy, Wolf, Life, figures or the repetition of life and figures, you have this systematic shift downwards. It's not dramatic, but the R-square are a little bit lower with the movies of movie 10 than they were on the Friends episode. And on the other graph down there, what you have are the different tasks of HCP. So again, the two first distributions are Friends season 1 and 2 as a reference, very similar. Some tasks are almost identical to the Friends. Those two tasks in purple and in grayish are the resting state and the social task. But tasks such as gambling in green or motor in red are substantially lower, and even lower than were the movies from movie 10. So if we do this comparison of individual parcels against group parcels, on the left we have the graph I already commented from Friends season 2. The general appearance of the graph is identical for movie 10, and it is also identical for HEPTRT. But if we focus on the gains of R-square between diffumo and DIPAC, so diffumo being here and DIPAC being there, it's quite large when we look at Friends season 2. It's a little bit smaller when we look at movie 10, and it's even smaller for HEPTRT. So even though globally those individual parcels do generalize and encode the data well and better than group atlases, the benefits we get from there get lower for tasks that are more different. And in terms of our ability to encode better with individual parcellations and parcellation coming from other individuals, once again, we have our reference graph we already discussed in detail for Friends season 2. Same appearance from movie 10 and same appearance for HEPTRT, where the blue is systematically lower than the red for all of the subject, right? Remember, each column here is a subject. But here the gains are massive. Here the gains are not as big. And movie 10 is somewhat in between. So once again, when we compare intra-subject versus inter-subject embedding, we reach the same general conclusion, which is the gains are massive for the same context that was used to generate the parcellation and become lower when we move away from the TV show to the movies to task in HCP. So, conclusions from the work and next steps. Well, we had three main objectives, right? The first one was to extend the dynamical parcellation method to the full brain. So the algorithm from Bookdeer and DAL 2020. Well, the proposed method, DIPAC, is a simple algorithm, which is, for me, a very attractive feature, and it does scale very well, at least to the size of the NeuroMod data, which is one of the largest, if not the largest, individual fMRI data set currently available. Our second objective was to assess the reproducibility of those parcels across the different cognitive contexts that we had. Well, the reproducibility was really good to excellent, near perfect for about half of the parcel when we went from the season 1 of Friends to the season 2. And once again, those videos were distinct. It's just same type of actors, same kind of stories. When we move to different genres of movies, action movie with Born Supremacy, the animal documentary with Life, then we see awkward parcels that start arising that are not really consistent with what we had on Friends. And for the control tasks, such as moving your hand or gambling on a decision task, well, the parcels there were actually quite consistent with the one of Friends, but it would depend on the particular task. Social and rest, for example, are very comparable to what we have in Watching Friends. Moving your hand, not so much. Now, our third objective was to look at the homogeneity of our parcels and look how generalizable it was across context. Well, what we found is that the individual DIPAC parcels have higher homogeneity than group parcels or parcels generated from other subjects. So that definitely support the usage of individual parcels, and that's true across all the contexts. However, the gains in homogeneity were very clear in the context where the parcel was generated and became less attractive the further away we went from that context. So the R square were maximal in French season 2, then a bit lower in movie 10, then again a little bit lower in HGPTRT. In terms of the next step for this, well, we've got a parcellation that can be used in NeuroMod. We are going to keep seeing how it generalizes across new tasks, right? Like we are constantly acquiring data. That particular study was on 40 hours of fMRI, individual fMRI data. We'll have about 100 hours more by next year. Actually, in our alpha release, we already have dozens of hours more than this. So we're going to keep going. We also want to start comparing with other algorithms. During the talk, I emphasized that this is not really about DIPAC versus other algorithm. It's about an individual set of parcellation versus some group atlases. And I'm personally shocked by how convergent the patterns we get from this K-mean clustering are with what I'm used to see with independent component analysis. And I suspect sparse matrix factorization as well, which is the algorithm used in Diffumo. So I would very much like to understand more the convergence of dynamic parcels with more like linear decomposition methods such as ICA and sparse PCA. Now the API to evaluate our parcels as well as our parcels are going to be made public. So I'm hoping that other groups are going to jump in and start, using NeuroMod as a benchmark for individual parcellation and compare what they have with what we generated. Hopefully, we'll see what happens. Third and last avenue, I would like to establish best practices for group versus individual parcellation. So we've seen that the individual parcellation explain more variants, they're extremely reproducible, and potentially they're better to capture individual brain organization. However, the Diffumo group atlas performed really well, and really when we went in HGPTRT, where the parcellation didn't generalize as well, the individual one, then the gains from individual parcels were not obvious compared to group parcels. And so depending on the question, maybe sometimes if you really want to be able to easily compare brain graph across subjects, Diffumo would make more sense. So I'm hoping that in the future, we're going to be able to compare quite systematically group versus individual parcels to get a feel of how important that particular decision is when modeling brain activity with artificial neural networks. And that was all I had for today. And I'm going to thank you very much for your attention. And I will talk with you during the interactive session. Bye-bye.